denoising part. Today's session will be like all about the construction of a feature table. So the construction of feature table does not have very long commands. It is just that the files that we generated last time, that is the rep6.qza and the table.qza, we will be converting those files into a visualization format and we'll be summarizing it. So that is the main concept of feature table generation. So I will be sharing my screen now and just let me know if it is visible or not. Is my screen visible?
Yes, ma'am.
Okay. So
Yes,
now,
it's visible.
okay. So now we'll begin with the session. So basically, this part of that metagenomic series is basically we'll be studying and focusing on how the sequencing data is processed after denoising and how we'll be interpreting it using a feature table in time 2. So where were we in the pipeline? So the steps that we have completed includes the demultiplexing, followed by quality filtering, where we remove any low quality reads and any chimeric reads removal using data to and deepler. We have corrected the errors. We have generated ASPs and we have removed any chimeric sequences. So these are the steps that we have already completed. And now our sequence is clean. It does not have any errors. And it has been, all the low quality reads have also been removed. So the outputs that were generated after the pipeline that we ran, like all the steps, now we have two different files. One is table.qca and one is rep6.qca. So whatever denoising stats that came, so those we only required to interpret that which and all sequences, like how many sequences were removed or how many sequences like had error and all of that. So that statistics was only limited to there, just to know that the denoising has been completed. Now after denoising, the files that are more important to us is table.qca and rep6.qca. So table.qza is the file that contains our counts, sequence counts, and rep6.qza is the file that contains our actual FASTA sequence. Okay. So we'll be seeing and converting these files into a visualization file, and we'll check what are the results and what are the outputs that these files show. So now this slide is very important conceptually. So initially we were focusing on removing any noise that is present in our data. Now we will be shifting towards extracting a biological meaning. So all the stages that we had in the last sessions, where there we were majorly focusing on removing, but as we move forward now, the main purpose of our thing is to extract a biological meaning. So initially we had the raw sequences reads, then we converted them into ASVs. Those ASVs were generated by the by the step that is denoising. So after denoising, we got a particular ASV for like each sequences, and now these ASVs will be taken forward and will be quantified, and biological information will be derived from these sequences. So now what do we have after denoising actually? So what do we have is, first we have a set of unique sequences, that is we have ASVs, collectively ASVs. Sorry. So we have on, I'm sorry. So initially we have a set of unique sequences. Just excuse me, like I have a little like cold, so I might sneeze in between. So just bear with it today. So what are we doing? Like what do we have after denoising? So this is the set of sequences, that is the ASVs that we have generated from our raw reads. So these are the ASVs that will be taken forward. So the The properties of ASV is that it is highly accurate, and here we have focused on a single nucleotide resolution. That is, instead of generation of OTUs that I mentioned, each and every sequence is corrected if there is any error, and then each and every sequence is 100% reduced. That is, each and every base is looked into, and that is why we called it a ASV. And then each sequence will represent a biological variant because we have corrected each and every sequence properly, and it is not clustered now, like it is present in an individual entity. It is not clustered in a group or something where we analyze the biological relevance for the entire group, but here we are going to determine a biological relevance for each and every variant, that is each and every sequence. This will give us much more higher resolution as compared to any other analysis. So here now each feature will be giving us a real biological signal, not an approximation, because we are not dealing with an entire group. We are dealing with an individual sequences. So now why do we need a feature table? So if we just have sequences, they are useless, right? Because they are not organized. So what do we need? Like if we need to move forward, what should we sort out first? We should basically arrange these sequences in an order that it generates, like it gives us some meaningful output, right? If all the sequences are just clustered like that, that will not be appealing for us also to analyze and all of that. So arranging them, putting them in a proper organization is what basically we are considering as a feature table. That is, we'll have rows and columns for designated things. The main questions that we need to answer, that is why we are generating a feature table, is that which sequence is present in which sample. Say we have one feature, so what is like, how, first of all, how many times that feature is coming, so that needs to be tabulated. Then which and all sample contains those features, that needs to be tabulated. And how is it varying across samples? So basically in one sample, like in how many samples one particular sequence is present, that also is an important factor that needs to be considered. These three are the main questions that we generally want to answer when we're constructing a feature table. And based on these questions only, the further visualization files will contain the visualization plots that will be answering to such questions itself. So now what are these counts that I keep on talking about? So what do they basically indicate? Okay. So this is like one of the most important concept that I would like to tell. So this is basically the counts depend on what, how are these coming. So basically the reads can be coming from a DNA extraction efficiency, for by PCR amplification bias, and by sequencing depth. So these are the different ways from where the counts can be coming, and counts can be affected. So the thing is that one sample or one microbe can have multiple counts, because it is not compulsory that the entire genome can be sequenced in just one read. It is possible that it can be sequenced in like multiple reads. So counts does not basically are equal to the relevance or the relative abundance of a particular species. Say if you got 1,000 counts for species A, and if you got 10 counts for species B, that doesn't say that species A is more abundant than species B, because the counts are more. The counts here are telling the sequencing signal, that is species A has been sequenced much more in a better way as compared to uh, the organism B. So that is not at all, because this is a major misconception which like people do have, which I also had in my initial stages, that oh, if the counts are high, that means the abundance of that microorganism is high. So if I'm sequencing two microbes A and B, if A has more counts, that means A is more abundant. But that is absolutely not true. If A is more count, that means that A has been sequenced more. That is, the sequencing signal is more, not the abundance. So we shouldn't correlate counts with abundance. Now the next concept is the sequencing depth. The sequencing depth is basically defined as that how many total number of reads that we are getting from one sample. So the same thing that I'm saying, that if we have two organisms, say A and say B, if A has 50,000 reads, that means it is high depth, and B has 2,000 reads, that means it has a low depth. That means this organism has been sequenced thoroughly. Like even the smaller or any rare a feature or any rare taxa that is present in in that particular sample can be visualized properly. Whereas in sample B, this depth is low because the reads are not proper. So here you can so here you can identify the major, like overall the prominent taxas, not like very rare or very at the low level. So that is what is depending on depth and high depth and low depth. So this is what also counts. If a sequence is a higher depth, that means the counts are also more because the organism is sequenced, the sample is sequenced very thoroughly. Whereas here the counts are low. So this doesn't mean that they are like comparable because it has been sequenced more thoroughly. Rather, it is not happening that properly. So we need to have a normalization method where we bring both of them down to a similar level and then compare. That can be also a possibility. If sample A has these many reads and sample B is this many reads, sample B is more prominent and is more abundant because compared to sample A, because the abundance is not compared to the counts, read counts, or the depth. The depth and the counts basically tell that how well an organism or a sample has been sequenced and how well you can determine even the rare taxas in that, not the abundance directly. So now why are we summarizing? So before doing any analysis, we must check the quality of our feature table. So we need to see if there are any poor quality samples, if there are any uneven sequences, and to understand the structure of the data set. So basically how they are present. Without summarization, there is a possibility that we can do a biased analysis the way I told here. So here you will know the sequence in depth and frequencies and counts of the sample. And if you wouldn't have known and if you would have directly compared, then that could have led to a biased analysis. Because this is sequenced more. Obviously, there's a chances of this getting like more analyzed as compared to this because this has higher number of reads and there's a possibility that it can come and it can mask the lower sequence depth samples. So to prevent all this, there's a thing where we summarize the feature table, and this is the where they summarize the feature table, and then that reduces the
Ma'am,
possibility of our doing bias analysis. Yes, Manvita.
ma'am, it's Chaitra. Ma'am, can you explain that previous slide by giving some biological examples?
This slide.
Yes, ma'am. Yes, ma'am.
Okay. Some biological example. Say you are sequencing a sample, right, from soil. So there are certain microorganisms. Say, uh, I'll take two species. One I'll take Bacillus, and one I'll take Streptococcus. Okay, there will be multiple subspecies inside this Bacillus and Streptococcus. Say sample A is basically spanning the Bacillus ones, and sample B is spanning the Streptococcus one. I know that in soil Streptococcus, this is a hypothetical example. Okay, I know that Streptococcus is more abundant, but when I'm sending it for sequence, the depth that is coming for streptococcus sequencing is coming very low. Like I'm not able to identify any more lower subspecies organism for streptococcus. But bacillus has been sequenced very nicely, very well. So there are a possibility that it can happen if I don't summarize the feature table that bacillus might overtake, and then streptococcus can be like as a suppressed, that okay, you stay bro, I have a lot more reads, so you are not that prominent, so let me come up and let me get analyzed and visualized when the analysis is happening. But that's wrong, right? Because even though they are more deeply sequenced, but streptococcus is the one that is more abundant. That is, we know from theoretical studies, so we can tell the answers that okay, when it shows bacillus is more and streptococcus is less, that it is not actually true. But we know that it is like on the other hand where streptococcus is more. So this is how we correlated it, how I understood it, because I had a previous knowledge. But when we are doing a de novo sample, that is, you don't know what organisms are present, what are the already habitants of that particular sample. So at that time, to avoid this false results, we need to compare it with a normalized manner. That is, we can't directly compare it because the read depths are very different. So we need to bring it down to a normalization factor, and then we need to normalize, and then we need to look into them so that we can get a proper decision. Not the only concept that is important from this slide is that even if the counts and the sequencing depth is high, that doesn't give a possibility that those organism or those samples are more abundant. So you cannot draw a line if a particular sample has higher read counts and a one sample has lower read counts. You cannot say directly that one sample is higher, is more abundant as compared to other. This is just the sequencing. You can say that yeah, one sample is sequenced more better way as compared to the other sample. Is it clear?
Yes, ma'am. Thank you.
Okay. Okay. So now we'll be running the command. So I hope you guys renamed right your table data to .qza to table.qza, which I mentioned in the last session. That MV command, right?
Ma'am, for the outputs we got from the deep pipeline, we haven't renamed them, ma'am.
We will be moving on with the data to output. So those are the ones that I would like you guys to rename. Have you run those commands in the last session to rename those files?
Yes, ma'am.
Yes,
For the
ma'am.
data to files we have renamed. For deep we haven't renamed them,
Okay.
ma'am.
Yeah. Okay, that's okay. Because we'll be moving forward with the data to things. And if you guys want to work with the debuggler outputs as well, so you can run that accordingly and simultaneously in the as a homework. But in the sessions, we will be focusing on data too because I have been taking that from the pipeline. So I have sent the command. You can run this command in your terminal, open everything. Like one of you just share your screen and show me. And both of you run this command and we will see the outputs, what and all outputs came. Any one of you can share your screen. Are you guys able to share your screen?
Yes, ma'am. We'll share now.
Okay. Yeah. Can you show me or can you list me the files that got generated? So table.qzv file is generated when you see at the command. A table.qzv file is generated. So can you see a table.qzv file? Yeah. A table.qzv file is generated. Can you see here on the where your cursor is?
Yes, ma'am.
Chaitra, did you also get the file?
Yes, ma'am.
Okay, so initially we had table.qza, right? When we generated from the denoising command. Now we have, you can see in the command, right? I'll come back to my, you can stop sharing your screen. I'll come back to the presentation. So here you can see that along with table.qzv, we are using a metadata file. So here we are trying to make a biological comment on the sequences that we have got from after denoising. So that is why I told that there is a transition now from the raw reads and analyzing, removing any error and all of that from correcting the sequences to putting a biological meaning behind those sequences. That's why we are using a sample metadata file in our these commands. So now table.qzb file is generated. Now what and all does it contain? So it will contain a per sample statistics, that is total number of sequences and the number of features that are present. And then to represent that, there will be histograms and bar charts that will be present. There are even frequency plots that are interactive as well, that you can see how it changes when the sequencing depth changes. And obviously a summary statistics where mean, maximum, median, everything will be mentioned. So after you generated a table.qzv, now we will tabulate. This is a second command where we are tabulating the reference, that is a representative sequences. So we had a representative sequences.qza as well. Now we will be converting them also into a .qzv format. So I have sent the command in the chat box. You guys run the same command and let me know when once it's done. The output for this command should be a rep.seeks.qzv file. So basically this entire table generation, feature table generation is based on the conversion of those QZA files to .qzv files, so that we can visualize them and we can carry out, like take out a biological interpretation from that, so that we can move on to the further post analysis step, that is the generation of phylogenetic tree and alpha beta diversity analysis, all of that. So did you guys run the command? Sorry,
Yes, ma'am, we got the output.
you got the rep.seeks.qzv. So these are
Yes,
the
ma'am.
two outputs that is basically important for us after we do the feature table generation. So this is how the files are supposed to look. This is for rep.seeks.qzv, and this is a explanation. And these are all explanations that I have put in this. So now I will be moving on to the explanation of the results. So you can go and read through this when I'll share the slides. This is like a written explanation all together. So now I'll show you. Sorry. So this is what the rep.seeks.qzv file looks, the file that we just generated. So you guys can also open and see, but right now I'll just explain it to you. So now, did you guys get the outputs right? .qzv files for both of them.
Yes, ma'am, we got it.
So now you can just
Yes.
upload to that view KIMO, and
Yes,
then
ma'am.
you should get the similar outputs. Okay. So now, and this is the rep.seeks. So what, like, I just want to ask this question: what were you guys expecting from a rep.seeks.qzv file? I mentioned in the last session, right? Like what this file should contain.
Ma'am, sorry, can you come again?
I'm saying I wanted to know, like, this is a general how this file looks. So I have already mentioned in the last sessions that what I was expecting in this file, right? What output I was expecting in this file, particular file, and is this the same
Ma'am, in
output
the
that I'm
rep
getting?
sequence, we get the original unique sequence related to the sample.
Yeah, but there was one specific thing that I mentioned, that we will be getting the full FASTA sequences of the, that is the entire sequence of the ASVs. That is what I was focusing more on. That in the representative sequences.qzv, you will have all the sequences, that is, you will have all the FASTA sequences that you have generated after the denoising part. Okay. So this is how it looks. So for all the 34, for all, not 34, we had features, right? We had 34 samples. From those 34 samples, we got large amount of features, right, that we processed and everything. So for each feature, they have given one feature ID. And then for each feature, they have given us the sequences. This is the FASTA sequences. And we have truncated all the sequences at 120 base pairs. So the sequencing length of all the sequences will be 120 base pairs, right?
Yes, ma'am.
Does this file make
Yes,
sense,
ma'am.
right?
Yes, ma'am.
If you guys have any doubt or you're not understanding, just let me know, okay? Because this is important to understand.
No, ma'am, we are understanding.
Okay. So for each feature, they have given a sequence and then they have given the sequence length. So this sequence length is basically 120 because we truncated everything at 120. So everything is uniform. That is all the features that we had from 34 samples. Everyone has 120 base pairs ka length. So similar thing is here. So the total number of sequence count is 770. Okay. So this 770, the total number of sequence count is 770. The sequence count is telling the entire sequences that are present here. And then the minimum length is also 120, maximum is also 120. Everything you can see here is 120 because we have truncated the things at 120. That is why we are getting it as 120 lengths. And when everything is uniform, the mean, median, 2%, 9%, everything will come out to be same because all the 770 sequences that we have taken here, they have all similar length of 120 base pairs. So that is why it is going to show like this. Now, this was how our rep6.qzv looked. So we expected it to have the FASTA sequences. It had FASTA sequences for all of it. Okay. So now we'll move on to the next output, that is table.qzv. Okay. So this table.qzv will be telling us the statistics part, that is the mean, median, and whatever things that are like whatever sequencing count is coming. Sorry. So now it is telling the number of samples that we had is 34. Okay. And then the total number of features, that was 770. That is, these 770 features are the ASVs. That is the ASV sequences that we generated after the denoising step. And now the total frequency for these ASVs is 1,53,807. So this number is basically telling that how many reads are corresponding to 770 features. So one feature can have multiple reads. Okay. So that is what this is suggesting to us. So now we have, this is the total summary where we are getting to know the proper total statistics, where it is telling the number of samples, number of features, and what are the frequency of the features that are occurring. And all of the statistics is after we have done the denoising part. That is, we have cleaned all the sequences. Now here, the frequency per sample section will basically show how these reads are distributed along the samples. Okay, so these are the total reads that we got. So now how are these total distributed among the samples? So that it is showing. So the minimum frequency is 897. So this is going to indicate that I mentioned right now depth, right? Depth is basically the total number of reads that are coming from one particular sample. That will be considered as depth. If you are getting so many more number of reads from one sample itself, that basically suggests that the sample is more deeply sequenced. That is, it has more prominent, like it is more prominent that the entire each and every nucleotide or each and every base can be determined because the sample is sequenced very deeply. So here the minimum frequency sample is 897. So basically this is the minimum. So this sample will basically indicate to the lowest depth sample. Whereas the maximum sample that is showing here is 9820. That is, sorry, that is, it is showing that the maximum sequence, that is the maximum depth sequence sample, has a frequency of 9820. Then let's come to the first quartile, median, and third quartile. So basically, when you see at the median shows that 40 4010. So this is not as much. Obviously, median should tell the middle part. So it is not as much. It is not very highly sequenced or with a very high depth or a very low depth. So it suggests basically that the most the most samples fall in that particular range. And when you see at mean is also coming out to be 4500 something. So you can say that maximum samples that have been like have a sequencing depth that can be considered as a moderate sequencing depth because they're falling under the 4000 line, because the maximum was 9000 plus, and falling somewhere around 4000 will be considered as a moderate length. And that is the mean. That means the maximum number of samples will have that sort of frequency. So you can consider that most of the samples that are sequenced contains a moderate sequencing depth. Now when you come to quartile, we can see in quartile 1 it is 1838, right, 1800, and whereas in quartile 3 it is 7000. So this is basically indicating that the 50% of samples, they fall within the range where that fall in the moderate part of the range. So now we can see here is there's one thing that you can determine from this particular thing is that in the first quartile, it is showing as 1838. Whereas in the third quartile, it is showing as 7038. So this will basically tell that there is some uneven sequencing depth that is happening across the samples. The same thing can be visualized in the histogram as well. So here you see that is the frequency per sample, and here the number of sample. So when there are initial number of samples, that is the maximum till 40, you can see that the frequency is lower for the reads for all the samples. Okay, there are very few samples in the end that have a high frequency. But there are, you can see there are two peaks here, peak number 1 and peak number 2. That is what this quartile is representing. So for this particular quartile 1, it is showing the mean as 1838. So you can see that the maximum number of samples that have a low frequency are falling in the initial ranges here. So that is why Q1 is showing as 1838. But when you see here, again, there is certain set of samples not as high as initial part. They are also showing some frequency that is like high frequency, but obviously the number of samples are not as high as the initial data that we got. So here we are getting basically two analysis where we are getting an uneven sequencing depth where we have two different peaks. For one, it is a little low frequency per sample, and that is present for maximum number of samples. And here the frequency for the samples is a little more compared to the initial, but the number of samples is not that much. So basically there are set of samples, that is maximum set of samples have a frequency of around 2000 something, where we are getting for the from the first quartile. And for fewer samples, say around 8 samples, we are getting this frequency of the samples around 6000 to 8000, that is falling between the third quartile. So these are the two uneven sequencing depth that we are getting, and then we can see from the histogram itself. Is this part clear what these table and graph are telling?
Yes, ma'am.
Okay.
Yes, ma'am.
So next it is telling, the next part here to understand is frequency per feature. That is, I told, right, there are 70 features, right? 770 features. So this was all about frequency of per sample, like how many counts or how many reads are occurring per sample. And now here we are going to see how many reads are occurring per feature. Okay. So the minimum frequency is 2. So this 2 says that a minimum of 2 features, that is for one feature, minimum of 2 reads are present. This is the minimum part. And the maximum you can see is that 11,373. For one feature, there are presence of 11,373 reads. Now you can see, right, this is more prominently and it is so highly dominant because it has been sequenced like in a very great depth, whereas some features are not sequenced in a very highly depth manner. And you can see the mean is nowhere near or half to maximum frequency. That means there will be some one or few features that will have a very high frequency count for the reads, but major samples that we will be looking forward will have a mean of that they will be consisting of around 200 reads per feature, and similarly the median comes up to 23. And similarly, it is shown in the graph when we know that the mean is also, and the here you can see the mean is much higher than the median. The median is very low. Median will be telling about the frequency in the middle, right? But the mean itself is very low. So that means you can see here that the graph is a little skewed, and it is little right skewed. So meaning, what does this mean? That there is only a few highly abundant features that are pulling upwards. So there are only like, and as I mentioned, that there are very few that will have that much high frequency, and their maximum are like in the lower range itself. So because of these higher things, that is why the graph looks like this. We cannot just ignore it and all. So that is why it is a little skewed, and it is pulling everything upwards here. So this is a pattern. So this is a pattern that can be seen depending upon this table. Is it clear?
Yes, ma'am.
Yes,
Okay.
ma'am.
So this was one part. Next is this interactive sample detail. So now these are the 34 samples. So these are the feature counts. These are the frequency for like basically how many reads are present for each sample. So for say this sample, the frequency came out to be 9,820. So you can see that this sample has the maximum sequencing depth, right? Because it has the maximum number of reads. So here, this is an interactive bar chart. So here you can see that when the sampling depth is kept to 0, these all indicate the 34 samples. Okay, all the 34 samples. So when the sampling depth is set to 0, that means that there is no subsampling happening and there is no filter applied. Therefore, 100% of the total sequences across the samples are retained. Because if we are just putting like, if there are 10 students and all the 10 students have different number of chocolates, say 1, 2, 5, 10, all of that. So if I say that all the students who have 0 chocolates stand in one line, nobody will come out, right? Because everyone has some or the other chocolate, even if it is as less as 2, but nobody has 0, right? The similar thing has been focused here. So when you say that sampling depth is 0, that means obviously every sample has been sequenced, right? It has some or the other sampling depth. That is why 100% of them is showing that, yeah, everyone is present. When you increase the sampling depth, say you put it to 2334 here. So all the samples that have sample sequencing depth less than 2300 have been highlighted in red. So, and similarly, they have been removed from here as well. So if you count, it will be the similar number of sequences that have been removed. So you can filter out the sequences that have a lower sampling depth. And you can put a threshold and you can just remove those that have a higher sampling depth, whatever you want to put for. So you can see, right? If I keep on increasing it too much, see if I increase it to the full, that is the sampling depth I've kept here is 9820. So see, you can see 9820 is the maximum that we can go. So if we put 9820, it will only filter out one sample because that is the only sample that has this many reads. So you can just play with it. You can move the bar and you can check if you change the sampling depth, how does the bar graph change, and how does the frequency and how does the sequencing change. So the samples that have a lower sequencing depth of what you put here will be removed. Only those will be retained that have a higher sequencing depth. Is it clear?
Yes, ma'am.
Yes, ma'am.
Okay. So now we'll move on to the feature details. This is the last part of the analysis here. So in this, basically, you are telling that how many specific features, like these are the unique, these are for features, okay? So these are those 770 features. And these features have a unique feature ID, okay? Each feature has its unique feature ID. So the frequency column basically shows the total number of reads that are assigned to that particular feature across all the samples. So here we were working with samples. You saw each sample has 9820 reads for this particular sample. And these reads could have come from obviously different features, right? So here what we are doing, here we are segregating everything based on features. So how many reads are corresponding to this particular feature? And this feature, it's not compulsory that it just occurs in one sample. This can occur in so many samples. So here it is written here. So this feature has a frequency of reads of 11373. Hmm? And in how many samples this particular feature is occurring? It is occurring only in 13 samples. How many reads, how many features did we have total? Sorry, how many samples did we have total? 34, right?
Yes, ma'am.
So out of 34 samples, this particular feature is present in 13. And the number of reads corresponding to this feature is 11,373. Now, similarly, you see, if you check out, so this particular feature, that is the third number, you can see the reads that are corresponding to this feature is 8,000 something. And this feature is present for 25 samples. So this feature can be characterized as one of the most prominent and prominent and a dominant feature that is present. It has a good number of reads also that are featuring it, and then it is present in the maximum number of samples as well. So this basically tells about, this table tells you about the feature detail, that is which feature, how many reads are corresponding to that feature, and in how many samples those features are present. Is it clear?
Yes, ma'am.
So these are the outputs that you get from repseeks.qcv and from table.qcv. These are three subparts. I've explained all of them. And I have pasted pictures as well, and I have also mentioned a written explanation. So just in case if you guys find difficult to understand again, so you can just read through it. And if you guys have any doubt, like if you don't understand when you do the revision, you can just let me know. I will explain it again. This is it for today's session. And this was all about feature table generation, construction, and then analyzing the .qcv files. So do you guys have any doubts?
No, ma'am.
Yes, Chaitra?